In this lecture, we are going to consider receptors functioning that results in action potential generation, conduction of action potentials along the nervous fibers, and finally, the way the nervous impulses are conducted between nervous cells and other excitable cells through special contacts, synapses. This is lecture part 1, where we consider receptors and nervous fibers functioning. Receptors are the primary initial source of action potentials generation in the nervous system. The receptors perform transformation of the various external and internal stimuli action into nervous impulses, which are similar to action potentials. And these impulses then can be sent to the central nervous system, and then they are conducted between neurons within complex neural networks of the brain for analysis and integration of information. And then impulses can be passed to the muscles or other excitable tissue cells to provide the body response. Receptors, sensory cells, receptors, exist in two basic types. They can be pseudounipolar afferent neurons or specialized sensory cells that contact the afferent neurons. Here you can see both cases. Case A, pseudounipolar afferent neurons. Here you can see only the nerve ending, while the body of a neuron is located somewhere distant to the right side, closer to the central nervous system in the spinal ganglia. This nerve ending has the membrane with necessary channels to produce depolarization in response to stimulus, and then channels for generation of action potentials. So under stimulus, receptor membrane depolarizes, and this is a receptor potential. This one, for example, is too weak to produce action potential, it's subthreshold, but let's generate the greater one, and then you see this receptor potential, let's assume that this one is enough to be converted into action potential on the membrane of the same neuron. And when it's converted to action potential then is conducted along the fiber to the central nervous system. So it's one and the same cell, just its nerve ending generates impulse, and the same cell conducts this impulse further to the central nervous system. These are simply arranged receptors, but much more complex are the receptors of the other type, which are specialized to detect specific stimuli, such as, for example, the sound for the auditory sensory system receptors, hair cells, the light photons, which can be perceived by receptors of the visual system, rods and cones, olfactory receptors that respond to different molecules. In response to stimulation, they just stimulate nerve ending by release of transmitters. So when a receptor potential is generated by such a cell, then this receptor potential makes the cell to release the transmitter from vesicle. Vesicles come closer and release their transmitter to the direction of the nerve ending. And these molecules bind to receptors of nerve ending, and action potential is generated as a result in the nerve ending, in the membrane of the neuron. And then it's conducted along the neuron. So many complex sensory systems use exactly their specialized sensory cells. Let's consider what is a pseudounipolar afferent neuron, and we compare with the central multipolar neuron. Pseudounipolar neuron has the receptor portion located somewhere at the periphery, for example in the skin, and the body of the neuron is located in the spinal ganglia, just behind the spinal cord, nearby the spinal cord. It's called pseudounipolar because it can be translated simply as false unipolar. You may notice initially it has something like one process, but it's not really one, false one unipolar. And it's then branched into two portions, one peripheral branch and one the central branch. And direction of conduction is shown here by arrows. So it starts on the periphery, and then impulse is conducted along the fiber, it passes by the body of a neuron, and finally goes to the central portion of the central process, and finally it excites the neuron that contacts this one neuron somewhere in the central nervous system. Multipolar central neuron has just one axon and it's not divided initially. Of course, in the periphery, it can be branched numerously, up to a few thousand even, of branches. But first axon is the only one, and the other processes that make it to be multipolar are dendrites. And this body is located inside the central nervous system. It means somewhere in the spinal cord or in the brain. Afferent sensory neurons send their impulses up to the central nervous system. This is just a picture that shows that it goes upward from periphery to the center, spinal cord and brain. Receptor potentials. Let's observe how they are generated. In most cases, receptor potentials are depolarization with just a little exclusion. And this is a change, depolarization of the nerve ending membrane or sensory cell membrane potential, which occurs under stimulation. So if you make a figure, it will look like from resting potential level, let it be minus 60, and threshold level, let it be somewhere between 40 and 50 minus, so it will be, for example, minus 45 millivolt. And then upward will be less negative values, while downward more negative values. And this is a resting membrane potential. And now, under stimulation, receptor potential may be generated. This receptor potential, as you can see, does not reach the threshold. So it's sub-threshold receptor potential. It does not bring excitation. It doesn't result in action potential generation. Let's make stimulation with stronger force. And this stimulation, as you can see now, produces receptor potential that reaches the threshold. So this potential reaches the level from which generation of action potential begins, threshold. Also, with stronger stimulation, receptor potential may be higher. It depends on the force of stimulation. It proportionally increases its amplitude in response to higher and higher force of stimulation. Receptor potentials. They are depolarization. Receptor potential that reaches the threshold and it results in action potential generation. Let's observe now again these three cases of weak stimulus which produces subthreshold receptor potential. You see, it doesn't reach the threshold, and as a result, no action potentials are generated. So the central nervous system doesn't receive any signals about this stimulation. But when stimulation reaches the threshold, receptor potential reaches again, and action potentials are generated. Under threshold minimal necessary force of stimulation, action potentials are generated, but their rate is low. Then, any higher than threshold, suprathreshold stimulation makes the receptor potential to exceed the threshold, and in response, in the period when we are above the threshold, action potentials are generated of the same amplitude as usual, but rate of potentials is higher. And this higher rate gives the information for the central nervous system about the force of stimulation. So, receptor potential itself responds according to the law of force. The higher the stimulus force is, the higher receptor potential amplitude is as a result. So this is type of encoding of information about the force of stimulation by the way of the analog encoding. Also, simply it can be called amplitude encoding, because exactly amplitude changes simply proportionally to the force of stimulation or analogically to the force of stimulation. Let's see, here is the lower portion of the previous figure, and you see stimulation force increases from first until the last stimulation. And in parallel, similarly behaves the receptor potential amplitude. Amplitude also proportionally increases dependent on the force of stimulation. This is classical law of force. The higher is force of stimulation, the higher the amplitude of response is. This is law of force. So this very simple encoded analog encoding then should be changed into the other type, because action potentials have constant amplitude. Their amplitude cannot be changed. And we cannot encode in force of stimulation by this parameter anymore. Let's observe action potentials. They follow law all or none, if you remember from previous lectures. The higher the stimulus force is, the higher action potential frequency is. This is type of encoding of the force of stimulation by the frequency. This is discrete encoding or simply frequency encoding. Let's see now the example. This example shows the action potentials of baroreceptors, arterial baroreceptors that respond to arterial blood pressure. So mechanical pressure acts on receptors, and in response receptors generate impulses. And you may observe that force increases in millimeters mercury in the lower line, and then you can observe very low pressure, 40 millimeters mercury, does not produce action potentials. It's subthreshold stimulation. But when beginning from, for example, 60, according to this picture, 60 millimeters already gives action potentials, but frequency is rather low. 100 millimeters mercury, higher frequency. And progressive increase in force of stimulation, which is mechanical pressure, hydrostatic pressure, the higher becomes frequency. In subthreshold stimulation, no impulses, and action potential is present in its full size, full amplitude, in case of any threshold and subthreshold stimulation. This is simply law all or none, or all or nothing. Amplitude is constant, you may notice it here easily. But the frequency progressively increases proportionally to the force of stimulation. In this case, this is the mechanical hydrostatic pressure. So frequency encoding or discrete encoding. This is higher force of stimulation results in higher frequency of action potentials, while their amplitude remains unchanged, as it should be always. So discrete encoding, this is the basic encoding of information for the central nervous system. And exactly due to high frequency or less frequency of impulses, we can judge about the force of stimulation. When impulses from periphery come with increasing force, we can feel, we have sensation of higher force of stimulation. Correspondingly with the decreased frequency, decreasing frequency produces the sensation that stimulation force decreases, and so on. This is the general encoding for the central nervous system. And also in technical area, the same types of encoding are used. Receptor potential and action potentials. Now let's observe in case when one and the same cell generates both receptor potential and action potential. And you can see receptor potential goes gradually increasing according to law of force, while action potentials generated just change their frequency, where the amplitude remains constant. So you can observe both types of encodings at the same figure now. Adaptation of receptors. There is specific state of specific capabilities of receptors. Not all of them equally able to produce adaptation, but many receptors. What is adaptation? Adaptation appears as a decrease of action potential's frequency in response to a stimulus of a constant strength. So the force does not change, and theoretically action potentials should come with equal unchanged frequency. But receptors that are able to produce adaptation gradually decrease the generation of impulses. This is true for receptors which do not monitor very important essential parameters of the body, and they deal with something less important, like for example the sensation of touch or odors. Let's observe. This is receptor potential, resting potential of the receptor membrane. Under stimulation, when stimulus is applied, the receptor produces action potentials. Stimulation is constant. Now it's switched off. And constant stimulation theoretically should produce action potentials with equal unchanged frequency. But you may observe the frequency decreases gradually. This is exactly adaptation. And receptors can be divided into fast and slow adapting. Slow adapting and non-adapting sometimes used as synonyms, because it was found that even receptors that considered to be practically non-adapting, they finally can adapt, but extremely slowly, for a few days, even not hours. So it's mostly they practically are non-adapting. So let's compare. This is case of non-adapting or slow adapting receptors. You see, in the interval of time when stimulus is applied, action potential's frequency remains unchanged. So, constant frequency of impulses. Such receptors we need for monitoring of the osmotic blood pressure, for example, of oxygen level in blood. So, oxygen chemoreceptors should be always non-adapting, as well as osmoreceptors. Otherwise, the information given to the important centers of the brain will be not true, and it's not possible, because immediately deviation of frequency of these receptors will activate the processes directed to prevent this decrease or any other deviation. So these receptors must be non-adapting, that respond to essential parameters of the body. But then let's consider what is the fast adapting receptors, what they do. Fast adapting receptors, in response to a constant stimulus without any changes of force of stimulation, produce impulses in the beginning with higher rate and gradually less and less. And finally, they can entirely stop their firing. These are fast adapting receptors, which decrease the frequency of impulses. And examples are olfactory receptors, the tactile receptors. Some very fast adapting receptors just produce few impulses in the beginning of stimulus action, and then practically they stop activity because it's unnecessary information that just produces additional information input to the brain, which is not necessary. Let's compare action potential and receptor potential. One type versus the other type. Of course, the comparison points can be many of them, but for main points, there are three of them. And first is law. Which law is subjected to the potential? Action potential is subjected to all or none law. It develops all with full amplitude, or does not develop at all, and there is no opportunity to generate half of potential or quarter of potential. Always potential is absent or present. While receptor potential follows law of force. With weaker stimulation, it can be small. With higher stimulation, it becomes higher in amplitude, and so on. Then, propagation, conduction. Propagation of action potential or conduction along fibers goes without damping. Damping is a specific physical phenomenon, a decrease of amplitude along the propagation with distance. And this damping is absent for action potential. And we'll explain later why it doesn't become decreased in size, because each point or each node of Ranvier produces potential actively itself. So there is no any decrease in size. While receptor potential, here you could see the conduction of action potential without damping. And receptor potential cannot be conducted to a distance. It becomes a little bit conducted to the nearest areas, but the more it goes further from the point of its appearance, it becomes less, and disappears practically. So it's not true conduction, just it goes for the short distance. Then with more distance becomes less, and disappears. So, but it's not designed to be propagated for long distances. It's normal property of receptor potential. So it's a dampened, decreased size, decreased amplitude of potential with distance from the point of its appearance. And last important point is summation ability. Action potentials cannot be summated. Let's recall. When stimulation is done, the action potential appears like this. But when we try to stimulate within the interval of action potential generation, like for example here, there is no any summation, because at this moment membrane is in its state of the absolute refractory period. No response. And when excitability is restored, new stimulation produces similar action potential and its amplitude remains the same standard as before. So we cannot summate two potentials at once because we can generate only one potential at the moment. It's similar like we cannot say two words at the same time. We should finish first word and start a new one. But as for receptor potential, it's able for summation. Look, if first stimulus produces such a receptor potential, which you can see. Now, in the middle of potential, when depolarization is still in progress, new stimulation produces additional opening of some more channels, and so repolarization does not occur, and depolarization goes upper. So there is a summation. You see, compare. Initial amplitude is less, and summation amplitude is a product of summation of the two, stimuli action and amplitude is higher. So this receptor potential becomes higher due to summation. So these 3 points are main points for action potential and receptor potential comparison. And last thing about potentials, I need to make clear which potentials generally follow law all or none and which follow law of force. And not only potentials. Due to potentials, some structures similarly follow one law or another. Let's consider first potentials. Clearly, the left side for all or none law will be occupied by the only one, action potential. And actually, this is the only one potential that is used for interaction between neurons, that is used for sending along the fibers, and long distances can be covered by conduction by action potential. It's a universal signal, universal brain language unit of information, and action potential is the only one really that follows all or none law. Potentials that follow law of force include receptor potentials and also potentials that we are going to consider in the next part of lecture. First, receptor potentials. You see, it's simply following law of force because you see they can develop different amplitude in response to different force of stimulation, the so-called gradual potential, which can be gradually more and higher and higher. And these are receptor potentials, but also the other potentials we will consider just in the next part of the lecture are postsynaptic potentials. Actually, the postsynaptic excitatory potentials, EPSP, look similarly. They also are depolarization cases. But postsynaptic potentials in inhibitory cells can be inhibitory, which means that they are hyperpolarization potentials. They similarly, just symmetrically, can be smaller and higher. And the amplitude, how much they go down to hyperpolarization side, depends on the force of stimulation and how much transmitters in response released into synaptic cleft. So these inhibitory postsynaptic potentials, IPSP, also are gradual and also follow law of force. These are main potentials. Then, structures. As a result of activation of some structures by single action potential, these single structures follow the same law, all or none. You see now the axon which conducts impulse action potential generated near the body in the axon hillock. And when this impulse passes through the axon, this axon becomes excited. Also, it can be not excited when no action potentials are conducted, and simply there is only resting potential. So also there can be only two states: excitation and action potential conduction, or absence of excitation, zero, the resting potential simply. So you may deduce from this that one nervous fiber, single nervous fiber, which is practically axon of the neuron, follows the same law as action potential itself, because it can conduct action potential or no potentials. There are no conduction of half potential. And finally, when this axon comes to the skeletal muscle fiber, it also produces similarly. If action potential arrives, it excites the muscle fiber and produces its contraction. If there is no potential, there is full relaxation, no contraction. And there is no intermediate state, half contraction or quarter contraction again. So the single muscle fiber also follows the same law, all or none, similarly to the action potential itself. But when we join the nerve fibers into great cable, which you can observe in the anatomy classes, you could see the nerves which are nerve trunks. Really, the nerve as a whole, here you can see the cross-section, consists of numerous microscopic axons. And if it's single axon, you cannot see it simply. And if you see this white thread in the body structure, these are cables that contain numerous nervous fibers. And within one nerve as a whole or nerve trunk, we can excite only part of fibers, and the other can be non-excited. And this percentage of excited fibers can increase gradually according to law of force. So this nerve as a whole or nerve trunk should be located to the right side where law of force structures are placed. Then single muscle fiber responds according to law all or none. But when we join into muscle as a whole many muscle fibers, the whole skeletal muscle fiber again, skeletal muscle again, can have inside excited and therefore contracted part of fibers, and the other part will not be excited. You see, muscle fibers in the skeletal muscle are not connected with each other by electrically conducting contacts. The only contacts are mechanically keeping them together, but conduction from fiber to fiber is not possible. Therefore, only fibers that receive impulse from neuron will become excited and therefore contracted. The other fibers stay relaxed. And so we can gradually increase force of stimulation and involve more and more fibers into contraction. So inside the muscle we can excite a different percentage of fibers, and this percentage increases with force of stimulation increase. Therefore the muscle as a whole responds according to law of force. And as you can see here, there is a little space more, and in this space somewhere we should place the heart muscle. And where it is placed? Let's observe. The structure of the cardiac muscle fibers is different from skeletal muscle. Doesn't matter that they are branched, it's different, but the main difference functionally is presence of contacts that allow conduction of impulses from cell to cell. These are contacts that are called intercalated discs. Actually, they are gap junctions, basically. And these intercalated discs, you can observe between cells, provide easy conduction from cell to cell. As a result, inside the heart muscle, it is impossible to excite part of cells. If there is excitation of part of cells, the other cells will immediately get this impulse too. Therefore, the heart contracts always entirely. Just there is division between the atrial portion and ventricular portion because fibrous ring is located between, but it's a specific division by connective tissue. But generally, if ventricles, for example, receive impulse, part of fibers only receive impulse, and the others simply become excited from their neighbors. And as a result, the heart always contracts entirely. All ventricles contract. And therefore, law of force doesn't operate here. There is the other way to regulate force of contraction, not through the force of stimulation. And actually, impulses that come to the heart are always standard, the same impulses. So there is no following the law of force, and therefore the heart moves to the left side. So it also the structure that follows law all or none. Now we see all the structures and all the potentials, division according to their subjecting to laws of excitable tissues, all or none, and law of force. Now let's switch to the nerve fibers. They are simply axons of neurons. You should remember that fiber is not something individual and separated from neurons. It's just a part of a neuron. But this part can be much longer than the neuron's body, and it may conduct impulses to the distant neurons or from neuron to the peripheral organs such as skeletal muscles. Here you can see the soma of the body of the neuron, with a nucleus in the center, numerous dendrites, and axon, which is single. And this initial portion of the axon that goes out of the body is the axon hillock, this little mountain from which axon begins. Actually, they believe that exactly here the density of channels for excitation is maximal, and therefore this is the place where excitation occurs easiest as compared to other areas of the neuron membrane. So it's a highest excitability area for neuron, and exactly here the impulse generation begins, when excitation comes to the neuron from other cells or from receptors. Here this is a cross-section of a great nerve trunk. So it's not cross-section of single fiber. You can see under one cover and one and the same, there are numerous fibers. Some of them are larger, and between them, if you look attentively, you may observe even much smaller fibers, which are not covered by myelin sheath, so they are unmyelinated fibers. So within one and the same large nerve trunk, there are many, many fibers. Let's observe how action potential is conducted along fibers. Conduction or propagation passing through the axon membrane goes. First, direction. From the central neuron, it starts at the axon hillock and goes to the periphery. As I already said, axon is always the only one, but finally, closer to periphery, the axon can branch numerously up to few thousand of branches. Of course, not necessarily always, it can be much less, but up to. So this conduction goes from the body to the periphery. In case of the peripheral pseudounipolar neuron, conduction begins at the periphery, and conduction goes passing the body in the spinal ganglia, and finally goes to the central branch, and then reaches the contact to the next neuron. Unmyelinated nerve fibers. Let's observe first the mechanism of conduction. These fibers are thinnest, and the velocity of conduction is slowest conduction. These fibers are used in the sympathetic division of the autonomic nervous system, where we actually don't need high velocity of conduction, because sympathetic nerves innervate internal organs, vascular smooth muscles, where their velocity, which itself is not very bad, is much more than enough. And observe, this is a cross-section of many fibers, and they are joined to a group by the single Schwann cell. Myelin sheath in the peripheral nervous system is formed by the Schwann cells. And differently from myelinated fibers, Schwann cell is present also in the unmyelinated. But you see how it behaves. One large cell protects and nourishes at the same time many, many fibers. Fibers here, cross-sected fibers, are shown by yellow circles. They are cross-sected. And you see many fibers are surrounded by processes, by pieces of the Schwann cell. And it's easy for one big Schwann cell to protect at the same time, mechanically protect the unmyelinated axons. So you see the difference is not absence of Schwann cell, but absence of myelin sheath that is formed by Schwann cell. And myelin sheath is greatly different from simply placing around of the fiber the portions of the Schwann cell. This is a cross-section. And protective cover. As usual, Schwann cells provide their functions of isolation, of nutrition of the fibers, but it's not myelin sheath, and therefore conduction mechanism here is very different. Velocity of conduction is from ½ meter up to 3 meters per second. As I already mentioned, that it's not very bad velocity itself, but as compared to other fibers, it's the slowest. So you may imagine that other fibers conduct much, much faster. Mechanism: how action potential is conducted or propagates along the unmyelinated nerve fibers. This propagation is achieved by repeated successive generation of action potentials in every point along the axon membrane. Every point. This makes big difference with the myelinated fibers. And just imagine that we take a little portion that is surrounded by this figure, and this little portion is enlarged, and now you see this. Let's assume that the axon hillock located is somewhere to the left, and therefore depolarization comes first to the left portion of this little piece of axon. And depolarization shown here in blue shows that exactly in this area at the moment channels for sodium are open, sodium enters to the fiber inside, and therefore charge is plus inside and minus outside. And what is important, here appears the area where the front of depolarization wave contacts the membrane in its resting state. So here you can see the contact of two opposite charges, plus and minus inside and minus and plus outside correspondingly. And as usual in physics, between plus and minus charges, immediately the current appears. And this current usually is shown from plus to minus normally. And correspondingly at the surface it goes from plus to minus in opposite direction, so that as a result you can see these currents are like circles. They are local circular electric currents. And exactly these currents produce depolarization of nearest unexcited area shown in yellow in this picture. And this depolarization opens sodium channels. And through open sodium channels, sodium starts to enter, and here, similarly to the previous situation for left side, similarly, sodium channels open and own potential is developed. You should understand that to depolarize the adjacent area, it is enough just to bring potential from minus 60 initial resting state up to, let's say, minus 45 millivolt threshold. If we reach only this threshold, then it's enough to produce opening of all the sodium channels present in this portion of the membrane. And this is already active process on. Through these open channels, sodium enters, and this area becomes depolarized. Previously depolarized area, left side, now shown in red because depolarization is finished there. Sodium channels are already inactivated, and potassium channels become open, and potassium starts to leave the cell, and repolarization process takes place. So, and the depolarization wave goes further and further to the right side. And here local circular currents develop now in the next area where depolarized portion of membrane contacts still undepolarized, non-depolarized. And the same local circular currents move along the membrane of the axon in the direction from center to the periphery. Actually, fibers are able to conduct in both directions. Just naturally, potential is generated in the beginning of axon, in the axon hillock, and therefore action potential travels only down the axon to the direction of the next cell, to the periphery of the axon. But if we take a piece like this one, actually if we stimulate the middle portion, conduction will go in both directions, but naturally it doesn't occur. So you see, resulting depolarization develops to the right side, while repolarization occurs in the previously depolarized area, and the earlier depolarized area now restores its resting potential. So this is a way to depolarize all the portion of the membrane, and it looks like the ring of excitation moves along the fiber from the beginning to the very end. And so every next adjacent point of membrane is depolarized to the threshold by the local circular electrical current. To the threshold, notice. And then this threshold is reached and depolarization goes itself because sodium channels become open. And this is generation of new action potential in every next points of the membrane. That's why it's rather slow as compared to others, because it takes more time to depolarize totally all the points of the membrane. Let's observe what's the difference for myelinated nerve fibers. I'm sure you know from histology that these myelinated fibers are much thicker and they have myelin shears, which is arranged in such a way that after each portion covered by myelin sheath, there is very small portion of uncovered axon membrane, which is called node of Ranvier. Axon hillock, then you can see these nodes of Ranvier. And this is internodal region. This exactly is the region that covered by myelin sheath, while in the nodes of Ranvier there is no covering. So it's simply membrane of the axon. And it happens very often, as you can see in the picture, that the diameter of axon alone is much less than total thickness of the fiber taken together with its myelin sheath. And the many layers of myelin sheath greatly increase the velocity of conduction. And actually there is the relationship. The thicker is the fiber, the thicker is the myelin sheath of fiber, the faster conduction goes. So this is an axon, this is the portion of myelin sheath, and this is the nucleus of Schwann cell, because the cell itself that produces the myelin, this is exactly Schwann cell. But here it behaves differently. It's wrapped many times. The thin layer of the cell wraps around in spiral way the axon, and many times it goes. Therefore, there are layers of membrane of Schwann cell, which is phospholipid, then little portion of the cytoplasm, content of Schwann cell, which almost entirely filled with a myelin substance that is lipid. So phospholipid membrane, then lipid, then next membrane. And these three layers are repeated and repeated and repeated. And with many layers of the lipids, there is a great isolation. You know, it's somehow it's similar to our isolation tape. Just the tape does not feed the axon, while Schwann cells do feed them. So it's isolation, nutrition function, simply protection, mechanical protection function performed by myelin sheath, and actually by Schwann cells that form the myelin sheath. And note this, the picture really doesn't show the ratio of distances, because if we do it proportionally to the real distances, to make distance between nodes even into different parts of the screen, it's necessary to make the thickness of fiber microscopic so that you cannot see it. Because notice, the open areas, which are nodes of Ranvier, make only 2 to 3 micrometers, while for the thickest fibers of type A, distance between nodes can be up to 1-2 millimeters. In some sources they can give even up to 3 millimeters. Just you may see that it's 1,000 time difference. So proportionally to show this, it will be hard, because it's necessary to make the node, one node in the left side, one node in the right side, and the thickness finally will become for the fiber so little that you will not simply see this. So that's why conduction goes so quick. But let's see how it goes. But first, how myelin sheath is formed. This is a picture that shows gradual wrapping around, like Schwann cell, in the process of formation of myelin sheath. Of course, mainly this process occurs when brain is formed. If you remember this moment when thyroid hormones are of high necessity in the perinatal period. But in the cortex area, we constantly have remodeling of the connections between neurons, and myelination can continue, so it's a normal process. So Schwann cell should wrap around, just Schwann cell is used in the peripheral nervous system. So for the brain, it's different cells. Let's see next slide. So Schwann cell wraps around the axon many times, and finally the myelin sheath is formed. It's a lipid substance that is formed inside in the cytoplasm. Sphingomyelin, it's another longer word. Cross-section of myelin sheath. You see, this is true picture. It's now not schematical presentation, and you see how nice these layers are made of myelin around axon, and how thin these layers are. And here it's schematic presentation to the right side. Myelin sheath, nodes of Ranvier. You can observe this. and myelin sheath formation in the central nervous system. Here, not Schwann cell, but oligodendrocytes. Here, oligodendrocytes. As you can see here, the difference is that one oligodendrocyte may provide isolation by myelin sheath for many fibers passing nearby. So not a single Schwann cell forms the internodal region, but it's formed by one and the same cell for many fibers. And now finally, how it's propagated. How the myelinated nerve fiber conduct the action potential, why it makes it faster. This is again achieved by repeated successive generation, but action potential is not generated through all the membrane, only in nodes of Ranvier. And if you recall that nodes of Ranvier make less than 1% of surface area, maybe even 0.1%. So you understand how much easier it's to do, is to do the action potential. Of course, here for schematic purposes, the nodes are shown very large and internodal distance is shown very small. But in reality, you remember that the proportions are entirely different. Again, let's assume that the neuron body is located somewhere to the left. Therefore, conduction will go from the left side to the right. Nodes of Ranvier are extremely rich in sodium channels, fast sodium voltage-gated channels. And myelin sheath covers the portions of membrane which even are not excitable, no channels. And excitable portions are only nodes of Ranvier, but they are highly excitable. And let's begin from the left. Initially, without action potential, inside we have minus 60 millivolt. But when action potential is generated in this one node of Ranvier, it means that sodium channels are open there, and through sodium open channels, positive charges come into the cell, and we reach, in the moment of peak, we reach plus 30. And this is the moment shown nearby the sodium channels, that inside we have plus. This is moment of excitation, and it corresponds, for the figure of action potential, it corresponds to upper point, the peak point, plus 30 millivolt. And then compare to the next, still not excited, node of Ranvier. Their charge is minus 60. And this difference of potentials between plus 30 and minus 60 is actually equal to the amplitude of action potential, 90 millivolt. So this 90 millivolt difference of potentials is so great that it's enough to generate the electrical current for a long distance between nodes. And these local currents develop, shown here to the left side. You may see these arrows that show the current from plus to minus to next node, and from the plus on the next node back to minus the previous node. So these are also local and also circular currents, but they do not look like circles actually now, because they are much more distant. They don't excite the nearby located portion of the membrane, but the next node of Ranvier, which can be few millimeters more distant from the first node. So, but this is enough to first, it's enough to bring the membrane of the next node of Ranvier to the level of threshold, minus 45. It's enough, because there is 90 millivolt difference. And all we need is about 15, maybe sometimes 20 millivolt to depolarize. And then this membrane produces its own response. This node opens all sodium channels, sodium enters, and so again here we have entry of sodium up to peak plus 30, and charge becomes plus 30. And now this plus 30 makes great difference for next node of Ranvier, which still is unexcited. And local currents develop similarly here. And then it excites the next node of Ranvier, and so it goes on. Really, the 90 millivolt difference is enough to excite not only one nearby located node of Ranvier. They say that perhaps 3 or more can be excited almost simultaneously with little time difference. So that in experiment, when exactly one node was blocked, it doesn't stop conduction, because this impulses conduction clearly can skip one node, because these local currents are strong enough to excite even after node Ranvier, which is more distant, and perhaps some more. So it goes extremely quickly. First great advantage is that we need to provide entry of sodium and then later loss of potassium just in small areas of nodes of Ranvier, which make less than 1% of the total surface of the fiber. Therefore, much less energy should be spent for restoration of initial gradients. To pump sodium back out of cell and potassium back into the axon, it takes much less effort. And then, if there is no necessity to excite all the points of membrane, and excitation comes like with big jumps, and that's why it's called saltatory conduction. It makes it much, much faster. By the way, you should understand the term saltatory conduction. Of course, no one jumps here. It's not a jump, really. It's just the electrical current that excites, brings the charge of the next, potential of the next node of Ranvier to threshold. And then this node generates its own potential. So it's not jumping one potential like a rabbit can jump and jump. No, it's generation of new and new potential. It's closer to domino principle. When one single piece of domino pushes the next one, and next pushes the next and next. So it goes exactly like this. One action potential excites the nearest node up to the threshold, and it falls itself. It generates itself own potential, and then it goes and goes. And the velocity is extremely high due to this. And in this nerve fibers classification, there is a grades of velocity. But first, observe. Fibers are divided into three types, A, B, and C. But classification is much bigger because A type is subdivided into alpha, beta, gamma, and delta. And actually there are also other parameters, not only velocity, but also thickness. But I believe that it's enough if you remember that A alpha are the thickest and fastest. And then gradually both conduction velocity decreases and thickness similarly decreases. But velocities you should have an idea. And first also division between myelinated and unmyelinated. You see, these myelinated are type A and type B. But type B is present in autonomic nervous fibers only. The autonomic nervous system doesn't need such great velocity which we need in our somatic system to control muscles and to get information from periphery, from sensory organs, from receptors. And you see type A alpha, fastest and thickest fibers, are fibers that carry commands from motor neurons to skeletal muscles. Types A, beta, gamma, delta, various sensory fibers provide information for central nervous system. Type B is used in autonomic nerves, and type C also in autonomic, but exclusively in sympathetic nerves portion, and type C has no myelin shears, while the rest types do have. Let's see velocity. We'll start from down portion with minimal velocity, 0.5 to 3 meters per second, and velocity progressively increases. There is little interlap in between velocities for type B and last portion of type A delta, but generally it goes without overlapping. So 3 to 18 is for type B, 5 to 15 type A delta, type A gamma 15-40, then from 40 to 70 for beta, and from 70 to 120 meters per second. Actually, all these velocities are approximate, and you can find the classification with a slight difference, but it's not of greatest importance. Basically, you should understand that myelinated fibers conduct fastest and from at least few meters per second up to 120, with type A alpha fastest 70 to 120, and slowest are type C up to 3 meters per second, which is not bad at all if we consider control of vascular smooth muscles. They don't need high velocity of control. And last thing I'd like to show you about the content of. Before now we considered only the fiber's membrane, because all the electrical processes actually occur in the membrane of axon. But what is inside of axon? Let's recall. Inside, if we take a little portion of axon, inside under membrane, it contains numerous microtubules. And these microtubules serve as a rails for transportation of substances. Many substances should be transported from the body of a neuron to the end of axon. And sometimes substances must be transported back, retrograde. From body to end of axon, it's called anterograde transport, directly. And backward transport is called retrograde. So, of course, the anterograde transport is faster. And the protein responsible for this type of transport is called kinesin. It's clear from the Greek word kinesis, movement. So, you can see a schematical picture here where vesicles, like spheres, are carried by kinesin molecules. And these kinesin molecules actually make something like steps along the microtubules. The pathway is microtubules, the direction is shown, and along these proteins and structures of microtubules, the protein structure of kinesin produces progressive repetitive change of conformation, which really resembles the steps of humans. Step by step, it moves and carries large vesicles. Actually, vesicle can be much larger than kinesin itself. Let's see this real picture. Here you can see microtubules, and vesicle carried by kinesin is so large that it even shifts the position of microtubules. And kinesin itself is shown by arrow. You see, this is small molecule, but this small molecule carries this large vesicle in a direction, and it goes step by step. In our site, you can observe in progress this movement picture that shows how kinesin can transport different vesicles. But this you can see later from your site. Now let's recall again. We have already idea about how neurons and also motor neuron get information from dendrites, and due to stimulation that comes through dendrites and also directly to the body, the neuron may generate its own action potential in the area of axon hillock. Now we have idea how this potential goes, which phases it contains, and how it's conducted along the axon. Through the myelinated, as this picture shows, for motor neurons they always are myelinated, how it becomes conducted along from node to next and next node. But then, when it comes to the end, axon is branched numerously in the skeletal muscles for motor neuron. And minimal number of branches is up to 10, and maximal it's up to a few thousand of branches. It depends on type of a muscle. It's going to be discussed later in muscles lectures. But now it's important thing, how this action potential is conducted, how it reaches the nervous ending. It's clear, should be clear at least. And then area of conduction is a neuromuscular junction. And you see, these junctions are structures that should conduct the action potential from the membrane of the neuron nerve ending to the muscle fiber membrane. And this is also a rather complex process, which we are going to consider in the next part of the lecture, in the synapse, because neuromuscular junction is a subtype of synapse. And by now, thank you for your attention.